Thank you for joining again. This is going to be a session dedicated to Exemplar project. So from this session today, we'll be transitioning from going over the practical and the technical aspects of data science related to biomedical data and transitioning into a discussion of projects. So we'll go over several examples. Today we'll go over some oncology examples. In our next session, we'll go over some other type microbiology project examples, and then we will talk about how to structure and prepare for a project, collect the data, and perform the analysis. So I'll give a little overview of how you can start thinking about projects, especially if you're thinking about oncology projects in this session. So we'll talk about networks of cancer regulation, at cancer studies and studies that look at different processes in cancer that underlie some of the clinical challenges in cancer that exist today. And we'll talk about kind of the concept of precision medicine, modeling precision medicine. How do we actually think of precision medicine challenges in the context of data sets that are publicly available, and how to combine those publicly available data sets with data sets that are maybe proprietary or come from other sources. Before we go into our discussion, I want to point everybody's attention into our progress. So let me go to the website. So as you know, we track how everybody's progress is going. And we are looking at two aspects of progress. So we look at the activity on the server, how you work on different pipelines. And then we also look at activity that is related to educational topics, which is completing the different types of sessions or different types of courses that we have online. So you can go to the organization page and see here progress. So if you click right here on progress, you'll be able to see, and then you can also modify the date range. And we started September 16th. That was our first session. You can see sessions down here. And today, this is October 21st. So you can see how everybody's working on the different practical things, as well as some of the online content that we have on the website. I also want to point your attention to all the participants, so you can see some of the participants and their activity. And I would want to ask you, you can see that Sarah, for example, is very active and has been throughout this session. So I just want to encourage everybody to try and complete their profiles. It would be good for both our purposes, for the purposes of the program, but also in the context of this project part of the program. It's important because we would encourage you to try and work together with other participants of the program, so you can potentially look at how you can complement each other's skills and understanding of the different projects. So each project is typically comprised of a number of team members, as we will see today, especially in these bigger studies. And we will see how typically they include people that have a little bit better understanding of the subject matter. In oncology, a variety of different aspects of cancer progression and different aspects of treatment and clinical information is going to be important for those of you that want to prepare an impactful project. Some people are going to be technically more advanced, so more able to process the data, analyze the data, integrate the data. And then somebody is going to be also more knowledgeable or experienced with biological interpretation. So we'll talk a little bit about all of these aspects, but I encourage all of you to just take a look again at some of the participants of the program so you can connect with them and you can look at how advanced they are to kind of see who you might want to reach out to. Now, briefly, I also want to review kind of what we have covered up until now. We looked at the program itself and talked briefly about next-generation sequencing and omics technologies. And we looked at how standard tools could be used for processing the data and just preparing the data for analysis. We looked at summary statistics and statistical properties, how you can kind of visually and statistically summarize the data, look at different data properties, and prepare some kind of an overview of what is it that you are thinking of in terms of comparing different types of data sets or groups and things like that. We continue to explore how exploratory data analysis can help us make sure that we're not looking at artifacts and make sure that some of the downstream steps are going to work well on our data, including quantile normalization, log normal distribution, and things like that. Then we started looking at some statistical methods that include understanding of regression, understanding of factors in our analysis, and trying to use correlation and regression to kind of compare different things and gain insights about features of our data in the context of statistical significance. Then we continue to learn about visualization and network analysis, where we look at how we can look at interaction between different features and placing the data in the of a network. We looked at dimensionality reduction, how we can summarize the data and kind of extract the most informative aspects of the data. And we used that, we used principal component analysis to do that. And then we looked at data mining, so unsupervised analysis like clustering, and looked at classification. Discriminant analysis and classification that really helped us understand how we can select subtypes and understand feature selection in the context of a specific question. Today, we're going to transition into projects to talk about how all of these different methods could be used in the context of a study. So in the context of the study, typically we're going to be looking at projects. And these projects, some of them already were covered in previous sessions. So I'm going to just quickly review how these different biological challenges can be translated into a project. So one of the projects that we already looked at was modeling precision treatment of breast cancer using multiple types of cell lines and therapeutic agents. We looked at just one aspect of that. We looked at gene expression in the context of subtypes of breast cancer. But if you look at the full project, you will see that there's a second component there, which includes the phenotypes that were derived from testing, high throughput testing of different therapeutic agents using these subtypes of cell lines. So if you would like to continue and explore this project is available on the project page. Let's look at some of the other ones that you can learn in the context of what we've already covered in the program. Analysis of the changing immune response in cancer is a study that illustrates how factor regression analysis can be applied to gene expression data in the context of development, context of time series, where we looked at the impact of different types of tumor-associated macrophages and their development over time. If you looked at the transcriptomic courses, a couple of them use this project called Whole Transcriptome Profiling of Cancer Tumors in Mouse Patient-Derived Xenograft Models. So this data set is interesting in the context being able to study the microenvironment of a tumor, where you can look at relation between gene expression in the microenvironment and gene expression in the tumor itself. Then today, we'll talk more about deregulation of subcellular networks in cancer patients. So this project is an example of how you can collect data from multiple types of studies, multiple types of cancer, and integrate them together to understand biologically what's going on, what is typical of cancer itself, and kind of separate from how it works in the context of specific types of tissues, and also how the tissue aspect plays a role in how cancer develops. But we do have a lot of other types of projects that are either in the works or already available. So for any of these projects, you can feel free to reach out to us and we'll guide you how to find the data if it's not publicly available on the site yet. For example, we have this project for biologics production using Spotaptera frugiperda and rhabdovirus contamination, an interesting project in the context of biotechnology and production of biologics. We have projects related to neurodegenerative diseases, projects related to Ebola and viral infectious diseases. So we can look at, again, use of machine learning and data science to kind of understand what drives spread of these different pandemic or epidemic infectious diseases. And of course, you are welcome to start thinking about your own project, but I do have to mention that, your project will be something that you will have to coordinate with us, and I'll guide you through the beginning steps of that today. In the context of cancer, we'll be looking at these 4 examples today. So we already talked about modeling precision treatment of breast cancer in a bit of detail. But also, there are some other ones that we covered, like early detection of breast cancer biomarkers, and then some of the projects that I already covered in the previous slide. Now, data science itself is not necessarily focused as much on processing the data. So processing the data includes pre-processing, some of the raw reads, mapping them, and then quantification. So we did cover that in the first lecture, but this is typically related to the core bioinformatics and not necessarily relate to data science skills themselves. But the 2 things that are related are the exploratory data analysis, where we cover descriptive statistics, visualization, and normalization, and analysis, so how we can test hypotheses, how we can mine the data, and using different classification techniques. But there's an important other component that we won't touch on so much in this program, but we do have other programs that cover interpretation, where we look at annotation of, for example, genes, pathways, microRNAs, these networks of relationships, how we use gene ontologies and pathway analysis to interpret the biological meaning of what we find during the analysis steps. So in the context of this program, we'll primarily focus on exploratory data analysis and the analysis itself. The other 2 parts, how to prepare data for this analysis and how to interpret this in terms of biology or clinical, is really going to be another set of sessions that we typically cover in other programs. For example, one of the things that we're working on now is a specialization track on oncology bioinformatics. We already had a program during the summer where we covered the biological and clinical aspects of oncology and all the way to interpretation of the found genes, pathways, and mutations. But we will, in this session, cover a little bit of that, but not as detailed as the various aspects of different types of data and different types of situations that are covered in the specialization track. So Before I go on, I just want to make sure that everybody has had a chance to review the material we've had up until now. We have posted all of the video recordings of our previous sessions. We have some online materials that you can go through, and project data sets that are already available on the site are available for all the participants of the program. So we suggest that you can go through some of those materials, kind of evaluate where you are in the context of the whole program. And then once you kind of see what is possible and what could be done, you, of course, are thinking about your own independent. So if anybody has any questions on what we've covered so far in the program or what I've covered in this part of the presentation, please put your question in the chat box and I'll address as well. So, okay, are the interpretation sessions done earlier? Do we have a set of recordings of them? Interpretation was covered in detail. So I'll talk a little bit about interpretation for oncology projects today, but the sessions on interpretation are typically going to be around a specific data type, for example, gene expression or mutations or some epigenetic variation. All of those will have different sets of interpretation methods. And so they are typically covered in our data-specific programs. For example, Omicslogic Genomics is a program that we just recently completed. So there we focused on interpretation of mutation variants, copy number variation, things like that. And there we, of course, have some of those recordings for that session, and we will be expanding the genomics courses so you can learn about that there. Interpretation of gene expression is a part of the transcriptomics series of courses. Interpretation of epigenetics or epigenomic data is also in the epigenomic course set that we have. So I was just looking at those, and then if you have any specific questions, we can talk about that separately, or we can guide you to the right program that will cover that. Okay, any other questions? Feel free to add them to the chat. So in the context of oncology projects and a lot of the clinical types of projects, what we're trying to do typically is find the underlying biology of some clinically interpretable phenotype. So we have the disease symptoms. Those are on the top, on the surface, something that could be detected fairly easily. But becomes complicated as you try to understand exactly what role that biological process typically associated with a clinical outcome plays in a specific patient. So we can look at this type of a challenge by looking deeper at the underlying subcellular biology, where we have this type of high throughput data that we can analyze. So for example, we can analyze genomic, proteomic, transcriptomic, epigenomic, and microbiome type data so that we can understand how it all works together to produce a clinical phenotype. And then it also helps us to refine our clinical data, refine clinical phenotypes, but also it helps us to understand how to go into maybe earlier stages of predictive analysis, how it can help us identify targets for treatment, and how it can help us understand the underlying biology and study it so that we understand how to intervene. So again, I think we've covered some of this before, but I'll do a quick review. In the context of, for example, breast cancer, what we see here is a mammogram, right? And in the mammogram, there is this spot. So this is a clinical data set, right? This is a clinical test. A mammogram is a clinical test, so it's performed in the clinic. And one of the challenges is to say, okay, when we spot this growth and we don't know how to characterize it, how can we go about this problem, right? So one way to characterize this growth is by looking at the tissue, where it came from. Now, in an organ like the breast, there are many tissues that could give rise to a growth, to tumor growth. So we can look at classification. Classification really means that we are trying to classify, put a specific type, a name to the growth. We can see that a lot of this is driven by histology, by how we characterize the tissue where it came from. Now, a lot of times, these growths, these tumors, have some elements that are non-tissue specific. When they're non-tissue specific, we are looking at cell types. We're looking at how different processes in cells differ from the actual source tissue where they came from. And this is called deregulation, because once a cell differentiates and becomes a part of a specific tissue, it's not supposed to continue and differentiate anymore. So we expect it to behave in a normal, predictable way as all the tissue cells behave. But sometimes they don't behave that way. And so we can spot these alternative types of processes by looking at specific genes or proteins. And so, for example, we can look at separation of different tissue into, of different tumors, separation into classes that are non-tissue specific. So for example, triple negative breast cancer, HER2, which is an amplified expression of the Herceptin, or of the HER2 protein, luminal, etc. These are going to be found in a lot of other types of cancers, but we are trying to do this by detecting a specific protein that could be found using immunohistochemistry, where we take an antibody, and it binds to a specific protein. And if it binds, we can then say that it's present, it's overexpressed. Now, unfortunately, this method, even though it's widely used in the clinic, is not very effective for several reasons. One reason is because we just don't have so much tissue. So continuously staining the tissue actually we use that tissue anymore, right? So we lose a lot of tissue. And so biopsy is a painful and invasive method of testing. So it's not used very often, unless needed. So it's impossible to get all the information we need just from looking at that. The other challenge is that we only have several proteins that we know for sure how they impact a clinical diagnosis, or how they might impact our ability to treat a patient. So that's why the rise of gene expression really gave rise to clinical panels that can help us in a much greater detail understand a classification, again, the problem of classification in the context of multiple players. So here you can see how PAM50, which is a clinical panel, helped us understand which subtypes of breast cancer are present. Now you can see that a lot of this is very similar, but a little bit different. And so that's the process that we're in, the process of refining the molecular subtypes of cancer based on tissue specific, organ specific, right? Organ specific would be breast cancer. Tissue specific would be, for example, lobular. Or now we're talking about molecular subtype, for example, luminal or basal. How are these tests or how are these methods useful in the context of our interaction with the clinician. So the first way that this is useful is by looking at survival prediction. So we're really looking at something that we can measure. So here you can see an example of a 10-year history of many different patients. And you can see how, based on something that you measure, for example, in year 3, will give you a very clear prediction about something that will happen in year 10, right? So that's the predictive concept here, that we can take some measurement today and so what will happen in 5, 6 years from now. Now, the earlier we can go, you can see right here, the separation is not very good. And because the separation is not very good, our predictive power isn't going to be as good. So what we're trying to do is we're trying to find genes in this example, which is a panel of genes. And you can see that these are actually different panels. A PAM50, which is a panel of 50 genes, out of which only actually 35 are the predict, and the other ones are housekeeping. So they just help us determine that we're actually performing a test on something that is biologically standard. Oncotype DX, a different panel. This example applies also to breast cancer. So you can see how this is a very important aspect of our ability to measure gene expression and predict clinical outcome. What kind of clinical outcome? Well, we're looking at survival, of course, which is RS, rate of survival, but we're also looking at rate of recurrence. And rate of recurrence means the tumor will come back. Now, not always does it come back as the same tumor. So here we also have an aspect of treatment, because a variety of these treatments like chemotherapy, radiology, they can actually cause cancer because they also cause mutations, because they also interfere with the regular cellular machinery. So the second aspect of the use of these panels is to select treatment. And so when we select treatment, we have the standard therapies, surgical therapy, right? So for example, one of the questions might be, how much surgery is needed? Do you need to remove the whole organ, a large part of the organ, or just a localized surgery? In that sense, it's important to stop the growth and stop the metastasis of the potential tumor and be able to get everything that might be acting in this cancerous way, to remove all of that tissue from the patient. A lot of times what is used is radiotherapy to kind of stop the growth, right? So if you apply radiotherapy, you don't apply radiotherapy to the whole patient, you apply it to a local, very specific region, and there you stop the growth, stop the progression of the cancer, and then you can sometimes even shrink the tumor, and then you can extract everything and hope that you got everything, right? But the danger, of course, is that radiotherapy itself can now cause a new process to start. So that's where an understanding of the risk level is going to be important for our ability to select the type of standard treatment. Now, chemotherapy, as you probably all know, is dangerous because it affects a lot of other cells and not just the cells of that tumor. So another kind of paradigm shift in the context of oncology is targeted therapy, right? And what essentially is targeted therapy? Targeted therapy means that we can target a specific group of cells. And so we can look, for example, at how do we differentiate just tumor from all the other ones and select processes that are relevant just to the tumor growth so that we can not affect any of the health cells. Now, that's a very challenging thing because, of course, cancer cells and normal cells all grow and divide. And so how do you separate between them is a big question, but it's really a question of biomarkers again, right? how can we look at biomarkers of just the tumor and find a treatment that will only target that tumor uncontrolled proliferation and growth and signaling pathways that we can stop that tumor from growing. But those same biomarkers actually work together with the other things that we looked at before, right? We can continuously monitor for different molecular subtypes, for rate of recurrence and rate of survival. We can look at also individual patients because they're not all the same. So a good example of this kind of a targeted therapy is Herceptin. Herceptin is an antibody that blocks a receptor, a HER2 receptor. And so this receptor actually is a gateway for a signaling pathway. And the signaling pathway, you can see the signal, actually causes the cell to proliferate uncontrollably. And so if we don't have Herceptin, we have this growth factor that comes in and binds to the receptor, and the signaling pathway is activated, and this process of uncontrolled differentiation happens in the cell. But if we block it, then everything stops growing, specifically in the cells that have an overexpression of this HER2. And that's why you see how it makes sense to measure for HER2 overexpression using immunohistochemistry. But unfortunately, not everything is so simple. For example, with Herceptin, we know that there's a problem of the changing profile. So if we use Herceptin, the tumor oftentimes stops responding. And that's because the nature of the cellular machinery is quite complicated. And so we can't continue and bombard all of these processes with the same thing all the time. And oftentimes we need an additional treatment to work together with some of these targeted treatments. So we block one pathway and then something else to block another pathway. And for example, here what you can see are some of the processes that are used, that are blocked to kind of effectively address this issue of Herceptin drug resistance. What you can see right here is actually how you can take population studies and develop an understanding of how this type of profiling correlates with these publicly available data sets, right? So for example, one of the things we'll be talking about today is in the context of all of this clinical use of some of these panels. How do you take a specific patient population, for example, let's say in India or in Italy, like here, and you say, okay, well, I have this population. Is this population represented in these larger studies that have given rise to these kind of, for example, panels or clinical outcomes? And a lot of times what you can see here is that it actually does not fully correlate with what's publicly available. So it's important that some of these data sets, they are comprehensive in a sense, but a lot of them have their own limitations. And a lot of these clinical studies are going to continue to evolve based on our understanding of biology, of how this works, but also based on the examples that we can look at. So for example, in different places, in different types of patients, with different types of treatments, and with different combinations of treatment, right? So that's why this is probably going to be an ongoing type of study that we'll see more and more split up into a diverse set of clinical indicators to consider as we look at the data and what it tells us. So the projects that we kind of covered up till now help us understand some of the biology behind what we looked at now, right? So for example, the PDX model project helps us understand the relationship between the tumor microenvironment and the tumor itself. And we can see a complex relationship between them. And so if we understand that relationship, we can design treatments that affect the tumor. And we can design treatments that affect the microenvironment around the tumor and work together on them to suppress tumor growth. And that's further explained with this tumor-associated macrophages process, where we see macrophages, which are immune cells that typically suppress tumors, but over time, the tumor can recruit them and actually make them into tumor-supportive macrophages. So understanding that process gives us an idea of how we can intervene specifically in those types of cells that the natural design of the human body actually already developed to fight these kinds of things, right? How can we not only suppress something artificially, but encourage something natural that already happens to suppress something harmful? And then here is actually a way to utilize different types of data sets that come from patients, but also that comes from cell lines, animal models, and how we can use our understanding of types of experiments that cannot be conducted on humans, but can be conducted on cell lines and animal models to really understand our ability to intervene and how to intervene more effectively. And so, for example, when we looked at this modeling precision treatment of breast cancer study, we can look at this publication. We just by reading some of the words mentioned in this kind of abstract, we can see how the types of things that we have looked at in the context of this program are actually going to be relevant to these types of studies. So let's just take a quick look at what it says. First-generation molecular profiles for human breast cancers have enabled the identification features that can predict therapeutic response. However, little is known about how the various data types can best combine to yield optimal predictors. Collective breast cancer cell lines mirror many aspects of breast cancer molecular biology, and measurements of their omic and biological therapeutic responses are well suited for development strategies to identify the most predictive molecular feature sets. So hopefully this program was effective to help you understand how to interpret what they are talking about. We're talking about molecular profiles, so we just looked at that now. We looked at identification of features, right? Feature selection. We looked at predictive analysis of therapeutic response. We looked at the various data types that exist. We looked at analyzing kind of the optimal prediction, right? How to evaluate our predictive power of a test. And we looked at, different omic and biological therapeutic responses and molecular feature sets, right? Just going through this program, goal for us was to introduce you to some of these things and give you practical examples of how to use them so that you can easily understand what these types of studies are trying to address. So now let's take a look at some of the methods that they used and some of the data sets that they had available to perform this study. So they used least square support vector machines. I believe we covered support vector machines in the classification session, but least squares is something that we typically cover in the multi-omics integration. Random forest, of course, we covered that. And then, of course, we use them to identify molecular features, which is feature selection. But from where? So they had 70 breast cancer cell lines and 90 experimental or approved therapeutic agents, right? So now we can see and combine omics data with other type of high throughput data and combine them together. Why do we need to combine them together? Well, what they found was that transcriptional subtype, which is subtype based on just gene expression alone, is a very strong indicator, right? Contributed strongly to response predictors for 25% of compound. But adding other molecular data types improved prediction for 65%. So in other words, that some compounds really work at the level of gene expression. And the gene expression patterns could be very predictive for about a fourth of these compounds. But for the other 3/4, you have other molecular data types that are strong predictors, right? So it's important to understand that a lot of these data sets, they have independent power to contribute to the type of test that we are trying to do. And that's why we see that no single molecular data set consistently outperformed the others, suggesting that therapeutic response is mediated at multiple levels in the genome. Response predictors were developed and applied to TCGA data and were found to be present in subsets of those patient samples, right? And now you're seeing kind of the translational value of these types of studies, where you can understand something, really collect the features that are in your data set, but then you also have to validate, and then you have to test your assumptions, your prediction, on another data set. And so they refer to the TCGA, which is the Cancer Genome Atlas data, to validate the predictive power of their test. So today we're going to look at TCGA and try to understand how we can leverage this data set so that we can study molecular features and their clinical indication power and their clinical application. For all of you that are starting to think about your project, these are the steps that you can think about as we go through some of these examples. So especially think of it because you need to clearly and in a focused way frame the problem. Very important to frame the problem, very important to think what is going to be this, this topic I'm going to put as the name of my publication. And when you frame the problem, you're also starting to think in terms of what type of data do I need to collect to understand, to answer the question that my problem highlights, right? For example, they were talking about modeling precision treatment of breast cancer, right? So treatment stands out here, right? Breast cancer stands out. And so we know that they want to collect data from breast cancer patients probably because those are going to be treated. So they need some information about treatment. They need some information about breast cancer. Now, what type of data from breast cancer? Their focus was on evaluation of different types of data that could be used as a predictor for treatment response. And obviously, they need the treatment information for that as well. Then you have to process the data. A lot of times we'll see how the data comes in processed, but it gives you a variety of options processed in different ways. So when it's processed in different ways, it's up to you to decide which one are you going to base your analysis on. So an understanding of the processing is also important there. You can't just say, oh, okay, well, I'll just pick randomly the first one, right? You need to do this with some logic and with some understanding. So hopefully you can understand the different ways the data processing is described. So you can say, okay, I'm going to use FPKM because such and such, or I'm going to use raw count, or I'm going to use the raw single nucleotide variation data in VCF format or in other formats. So you need to understand what format they are, what do these formats mean, what do these abbreviations mean. So that's why we looked at processing of data for analysis as well. Once you collect your data, in many cases, you're going to have multiple types of studies that were done in different labs, that were done by different people, that were generated by different instruments. And to combine that into a single data set that you can actually explore and understand is very important. And then once you explore and understand, a lot of times you have to modify some of it or trust some of it more than others because of your understanding of where that data came from. Once you've prepared the data, you will have to perform an in-depth analysis. And that's going to include multiple steps where you probably do some data mining, you probably do some classification, you probably combine them into a pipeline of doing multiple steps, and you will get repeated results that you feel confident about. And once you have that in-depth analysis performed and you've interpreted the data, you understand the biological meaning of what you found, you have to then effectively communicate the results of your analysis and get some feedback from people that are also looking at the same problem. So to help you start thinking about this process, we have this form. This is the form where you can submit this information, And so just briefly, what are we looking for? So we're looking for a problem statement, right? For example, I will take the breast cancer genome atlas collection of samples and analyze them for triple negative breast cancer patients and their age, okay? Something like that. Then you describe your project, okay? Why are you doing this? Well, triple negative breast cancer is the most diverse set of patients that have complicated phenotypes and really don't have an effective treatment strategy. So I decided to focus on the triple negative breast cancer patients, and I want to investigate the role of aging in the context of cancer, which are obviously related processes because of natural accumulation of somatic mutations. So that could be my project summary. Then I have to specify where is that raw data. So what kind of raw data did I use? Did I use prepared process data like expression tables, or did I take the raw FASTQ files? And I need to kind of explain that and probably provide some links. Reference publications. Why did I decide that this is an interesting topic to focus on? What are some publications? Who is the principal investigator? Again, if you're working in a team, you might play a role, but you might be working together with somebody else who maybe has a much deeper understanding of the triple negative breast cancer pathobiology. And so you might want to put that person as a principal investigator. Experimental design contact. Of course, if you generated the data or a lab generated the data for you, you can fill that out. But if you don't, just play not NA, not available. So that's, is it possible to get raw FASTQ files from TCGA? It is, but you have to get special permission. So you have to submit your project for the TCGA committee, first of all, to consider. Because of the private information, the FASTQ files contain some raw data directly from them, so they require for you to have proper infrastructure to analyze the data, has to be compliant with some of their regulations, and they have to approve your project. So yes, it's possible, but it's not as easy as just going there and finding FASTQ files. Okay, so let's look at the Cancer Genome Atlas, TCGA. And we look at TCGA from several standpoints. The first one is the growing use of machine learning, and we'll see a couple of examples. The second one is in the context of multi-omics integration, which we already saw up until now, but we'll see a little bit more why it's playing a role in analysis of this kind of patient-derived data. So first of all, about machine learning. Machine learning has been growing as a popular method to analyze these kinds of complicated and growing complexity data sets. So here's an example from one of the publications. This publication is also in the Dropbox link that you received for this session. And what you can see is that since the publishing of the Human Genome Project data, the rise of the publications that use some kind of machine learning technique, right? So it's important that a lot of people are starting to turn to machine learning to analyze this kind of high throughput, complicated data. Why? Well, because the patterns in this data are becoming more and more complex. And also you can see that from about the same time, there's also a continuous development in terms of the publications and also the patents that reference machine learning as well. So you can see that the use and then the results are growing in importance. So for example, here's a publication. Again, this publication is in the Dropbox link. How people use machine learning analysis of gene expression data, reviews novel diagnostic and prognostic biomarkers, identifies therapeutic targets for soft tissue carcinomas, right? So again, we see the same pattern. People use some method of machine learning, and you can see how a lot of times this is not going to be your standard approach. It's not going to be a random forest out of the box. It's not going to be clustering out of the box. people have to think now to adapt these methods to their understanding of the data and the challenges the data presents, and also the biological significance of a variety of different features that are used. So you can see that in a lot of these, some array of data is going to be used. For example, here you can see that they use the Cancer Genome Atlas and the French sarcoma group, right, that has a genotype tissue expression project. So combining two types of data sets together using, again, here you can see 3 groups of tumors that overlap in the molecular profile as seen with unsupervised T-distributed stochastic neighbor embedding, TSNE, clustering, and a deep neural network, right? So now they've combined a number of different machine learning methods. And then the 3 groups correspond to subtype or morphologically overlapping. So the morphology of those tumors is similar. And using a random forest algorithm, they identified novel diagnostic markers, right? So they first of all did exploratory data analysis, TE, clustering, right? That was their data mining part. And they wanted to find similarities of clusters by using classification, but it wasn't classification based on their annotation of clinical understanding of data. It was the interlinking of different clusters, right? So you can train a classifier on the results of your clustering. And then they used feature selection, random forest, to specifically look at, okay, which are the most important genes, which are the most important, right? We know it's genes because they're looking at gene expression data. And what were they trying to find? They were trying to find prognostic genes that are strong predictors of outcome, right? So again, now you can see that their conclusion is that machine learning algorithms are powerful new tools to improve our understanding of rare tumor entities, right? So they had a very specific focus, rare tumors. They used a number of different data sets, their own data set, the French sarcoma group, and the TCGA as a reference. They applied some machine learning from the data mine, from the unsupervised clustering and TC. And then they used feature selection to actually understand, well, which genes are responsible for this. And they actually try to do biological interpretation so that they understand, right? You can see reconstruction of regulatory networks. How is this actually working, the biology of this, as well as how predictive is the outcome, clinical disease outcome. Okay, so by looking at these different examples, we can learn about how to frame our own project so that we can make an impactful study analysis that will lead to some kind of exciting results. Another example, like I mentioned, so the first one was focused on machine learning. The second one is looking at multi-platform analysis of 12 cancer types. Right, so this is integration of data. And this is actually a publication directly from TCGA, where you can see that they are looking at pan-TCGA studies. And we'll look at a few examples more in the context of this lecture, because actually the TCGA data repository provides us with all of the pan-cancer publications that come out based on the TCGA data. So TCGA, what is it? Well, TCGA is a pan-cancer resource of clinical and omics data. And actually combines a lot of different information that you can find about different types of cancer. So you see different tissue types. It also gives you information on different genes, different mutations, and all of this data is searchable in different formats. So for example, you can see here the actual portal. So we take, let's take this link right here and we'll open it. And you can see how in this portal, you can actually easily find data that you need, but it's going to come in a very standardized protocol that is difficult to manage, right? So for example, if I'm interested in breast cancer, I can click here and start exploring the different types of projects. The different primary site is all breast, right? Then I have 2 projects. I have multiple disease types, genders, vital status, like dead or alive, things like that. And so now I can see how many do I actually have. I have 3,682 cases. So by looking at these cases, I can now refine my search and find all kinds of phenotypic information that I can study the data about, right? So I can now also see the different types of data that I have available for each one of the patients. A lot of patients to search through. For our purposes, a lot of times we don't know exactly what we're looking for, so we want to start with data mining. So how can we actually look at data mining? Well, you have here, this is actually an example of type of kind of important features that have been identified. So you see number of cases affected, and then these are copy number variation, specific information to gain and loss in copy number variation, right? DCN, as you know, is a gene. So I can look by gene and see which type of cancer and copy number variation of this gene, where is it playing a role, right? So a lot of different uses of this type of study. Also, NCI, the National Cancer Institute, provides us with the data itself, and let's look at what kind of data it gives us, and also it gives us a catalog of different other types of information. So it gives us the Cancer Genome Atlas for adult patients, and then also you have the Target Project for the pediatric equivalent of the TCGA. It provides the radiologic and pathology images, so you can actually look at images from the Cancer Imaging Archive, and proteomic data. So genomic data is going to be all the sequencing data that we talked about a lot in this program. Gene expression, genomic data, so DNA variation, epigenetic data, copy number variation, things like that. You have radiologic and pathology images, and you have proteomic data. And then also you have additional data sets that are not raw patient specific, but they're more conclusions. So based on, for example, somatic mutations in cancer, somatic mutations involved in treatment response and involved in cancer progression. Genomic encyclopedia DNA elements, for example, genes and isoforms would be gene elements. And then you also have the Cancer Cell Line Encyclopedia. So encyclopedia of different cell lines derived from patient that allows us to order a specific type of a cell line to study a specific subtype of cancer and things like that, right? As you remember, is in modeling precision treatment of breast cancer, we took a bunch of different cell lines and we needed to know clinical information about those cell lines. Now, the easiest way to look at the different publications that come out from the TCGA project is to look right here. They have, and you can also click right here on GDC apps, and you can see what they offer here, data portal and things like that. And so one of the things that you can see are publications, and you can search for publications in terms of different types of cancer, like breast cancer related publications, but also you can look at publications for pan-cancer studies. So that's what we're looking at right now. You can see how the TCGA data became a resource for data mining papers because it is processed and generated and processed in a consistent way. So you can easily create data sets to study these multi-omic and pan-cancer types of situations. Yeah, specifically, they even have a category called Pan-Can Atlas publications. So Pan-Cancer Atlas publications, where they look at different types of gene expression, you can see here, for example, supplemental data, right? You can just see different types of data sets that you can find for these specific pan-cancer studies, right? All of microRNA, all of RNA expression, all of DNA methylation. That's an easy way to browse your data, though, for our purposes, is going to be using another portal that we can actually use, and I'll give an example how to find it. So back over here, I have this link called FireBrowse. So FireBrowse is actually a place where you can more easily download the full data set that you're interested in analyzing. So for example, you can look at, let's find breast cancer, breast invasive carcinoma, BRCA, right? And now you can download all the clinical data or all the microRNA expression or the mRNA, right? So you can just download all of these data sets in bulk, and that makes it much more easy for studying these different types of data analysis techniques that we talked about in the context of this program, for example, clustering or classification. So let's take a look at one of these examples, how we can actually apply this type of a process to a specific type of a project. So this is an example, this is a publication, right? In Cell, you will have publications that come out from the Cancer Genome Atlas Consortium that actually analyze collections based on specific conditions, and then they publish all of the results in one review paper. So this is one of those review papers called Comprehensive and Integrated Genomic Characterization of Hepatocellular Carcinoma. And it talks about liver cancer, and specifically a subtype of liver cancer called hepatocellular carcinoma, which is from a specific cell type, hepatocytes. And they used, again, multiple types of omics data, DNA copy number analysis, DNA methylation, RNA, which is gene expression, microRNA expression, and proteomic expression also. And so you can see how they've identified several mutated genes, but they also looked at other ways of regulation, hypermethylation, for example. And finally, their goal was to provide an integrative molecular subtyping, just like we did with the breast cancer data, incorporating unsupervised clustering of 5 data platforms, identified that, etc., right? And they also developed potential therapeutic targets. So again, we see the same pattern: multi-omics integration, clustering, identification of similar clusters across multiple types of data, and then using classification to select top genes that could be used as potential therapeutic targets. And so you can see kind of a visual summary of this data right here. And here we have the link to the PDF of the publication, which is also available in the folder that was shared with you. So in the sense of this hepatocellular carcinoma, they have to give a reasoning of why they chose this, right? And as you look at this map, you can see incidence and mortality of HCC patients. And actually, worldwide, this is the 5th or the 6th most new cases per year worldwide, right? So 782,000 cases per year of HCC. So it's a pretty important area to study. You can see how it's distributed in different locations, right? And what is the type of we have to study this problem? What we have is, for example, from FireBrowse, or actually from this paper itself, you can download the clinical phenotypes. For example, you can see the clinical phenotypes here that talk about ethnicity, gender, year of birth, race, year of death, etc. And for example, if you look in more detail from this paper, you can also see hepatitis serology, BMI, obesity, and other factors that they have decided to be important to study hepatocellular carcinoma. So for example, if we look at the whole project overview, we can see that we have phenotype, we have different tissues. In this case, we just have HCC, different type of omics, and within each type of omics, we have different. So the question is, what can we do with this data? To help you look at this data, here are 3 example projects. One is, can we look at gene expression in the context of pan-cancer? So if we have cancer tumors arising in different tissues, for example, breast and liver, do they share anything in common? Or how is the tissue factor playing an importance in development of these tumors, in the differentiation of these tumors? Or you can take and study phenotypes. You can take a number of samples and look at the role a different type of phenotype plays in the gene expression, for example, or the microRNA expression of these different tumors. And another one, of course, that you can try is, okay, can I try multi-omics integration? Can I use partial least squares, combine 2 different types of data sets, and identify how they separate? Or can I take something like iCluster Plus and see how clustering would work on multiple types of omics data, right? So one of these examples is already provided to you in Dropbox, which is the 6 samples for pan-cancer. So please try and perform some analysis. I'll show you what that data looks like. It's going to be a number of samples that come from different cases downloaded from Firebrowse. So you can do it yourself, or you can look at this data here as an example. So you can see that this is LIHC, so that's liver cancer hepatocellular carcinoma. TP stands for tumor. So we have 3 LIHC TP, 1, 2, 3. 3 non-tumor, which is not taken from tumor, from normal tissue. And then we have BRCA, breast cancer, 1, 2, 3, and non-tumor, 1, 2, 3. So we have 6 samples from each tissue. And the question is whether you can find, is there a difference between tissue type, breast or liver? And is there a difference between normal and tumor? Okay, so that's essentially the assignment for you to try, or a helpful data set for you to try and play around with, right? And if you remember the types of analyses that we've done up until now, you can think of more of a data mining. Is there a difference? And then you can also look for a classification problem. Well, if there is a difference, in which types of genes? Which genes are showing the difference? So take a look at this data. This is something that you can practice with. Any questions about this so far? Okay, I know that we are running short on time, so I'm going to try and kind of finalize the presentation quickly, but if you do have any questions, please feel free to put them in the chat box. I'll be happy to pause and address those as well. So oftentimes, as you mentioned, you can also, so 3 things, right? So the first one is you can use different types of omics data, right? Multi-omics integration. Second thing we talked about was machine learning for specific type of data, for example, gene expression. And the third thing is really that in a lot of cases, we're looking at combining TCGA with other resources. So a lot of times it's using TCGA as a reference, another data site, database, or data set as your tree, for example, right? So what are some examples? Well, here we have, kind of building up on the same idea, characterization of gene expression profiles in HBV, that's hepatitis B virus, related liver fibrosis patients, and identification of ITGB1 as a key regulator in fibrogenesis. And then here you have another example, a pan-cancer compendium of genes regulated by somatic genomic rearrangements across more than 1,400 cases. So in a lot of cases, you will see how a data set, for example, TCGA does not have a lot of representatives outside of North America. So if I went to China, for example, which is what this is from, is a study of Chinese patients, could I study specifically HPV-related hepatocellular carcinoma and then see how is it different from TCGA, right? So those are the types of examples that you can leverage TCGA in, and sometimes it's a good resource and sometimes going to be limited. Another way, of course, to do this is here's a way to combine, for example, mouse animal or animal studies with human studies. And a good resource for that is the GenCode project, which is identify and classify all gene features in the human and mouse genomes with high accuracy based on biological evidence, and to release the annotations, right? So a good resource to study, for example, human tumors in the context of animal models. So these are some of the topics that I wanted to cover today. Specifically, how can we understand the clinical use of machine learning for this type of biomedical data, omics? We can combine different types of omics together to understand how they relate to a clinical phenotype. And what is a clinically relevant phenotype? Survival, recurrence, some kind of predictive analysis, or selection of therapeutic targets. And then finally, how to find and utilize different types of data sets, for example, TCGA, non-TCGA, how to combine them together for an effective study. And so with that, I just want to mention that in our next session, we're going to be looking at the same type of project, same type of an overview of different types of projects, but more related to microbiology. How can we look at, for example, resistance? How can tuberculosis? How can we look at Ebola as a viral infection that causes disease and spreads as an epidemic? How can we use machine learning in the context of those examples? Because we're going to be talking about microbiology projects, I also want to encourage everybody to take a look at our upcoming metagenomics program. So the metagenomics program, for those of you interested in microbiome, different types of analysis of microbial communities, individual strains, or diseases, clinical manifestations of those microbes, like viruses, mycobacterium, and things like that. We will have a program that's going to start in November, and we're happy to share the link. I don't know, Pratim and Mohit, if you could share the link for that program as well. We'll be discussing to find the data, how to process the data, and then how to build an independent project using this data to really understand metagenomics. So that's all I had for today. Again, team Bipsa, Mohit, if you can add the link to the metagenomics program, how to register for that, that would be great. And again, I want to remind all of you that we have already pasted a lot of the materials, including this presentation and the data sets and the publications that I highlighted in this presentation. And you have the links in chat. So please take a look at those as well. And I'm happy to answer any questions or take any comments. Okay, for Vamshi, in framing the problem, right? So framing the problem is very important because you want to focus on something that is novel and something that is relevant to study your data set, right? So before you even start with the data set and you say, oh, I just want to do something in breast cancer, you want to review some of the publications, for example, that we talked about today or in general on NCBI, and look at what are high value important questions that exist. How can I frame those problems in context of data, right? And then you go to selecting your data. So asking yourself a question, why do I want to study this? Why is this an important topic for others? What is the clinical relevance of this? What is the pharmaceutical relevance of this? What is the epidemiologic relevance of what I'm studying? So those are the questions you want to ask, more on the context of why, right? Why am I trying to do what I'm trying to do? After you've answered that question, you have a compelling story. I want to study triple-negative breast cancer patients because there's not an effective treatment and there's a lot of diversity. So there's obviously a need for better defined subtypes and new therapeutic targets. Okay, now you have your why. That's why you want to study what you study. Then you're going to transition into how. Well, what kind of data do I need to be able to address this question, right? And so I think that it would be good for all of us to review some publications that exist in the topic area that you have chosen, find that problem statement, and compile it in a concise way to be your project title. And that would be framing the problem. Does that make sense, Vamshi? Okay, what's the due date for the project? So it really is based on when you want to have your project completed. Just in terms of planning a timeline, it typically takes at least a month to do something with your project, depending on how much time you have, depending on how much you already know how to do, right? So from now, we have some time until the end of the program, but it's not that much time. So if you want to complete your analysis before the end of the program, I suggest that during this week is when you start submitting your project idea. And it will probably take a couple of times to refine that project idea until you actually start working on your project. So I expect everybody to have something already now. If you submit the draft, we'll talk about these ideas. And then by next week, everybody can start their analysis, and you'll see how a lot of these steps that we discussed today will play a role in how long it takes to actually get to something that you can feel is presentable and effective. Okay, any other questions? All right, great. Thank you, everybody, for joining today, and hope to see you again soon. We'll actually see you again this week, and we'll talk more about some of these microbiology projects. Thank you.